So I'm going to talk today about my favorite phenotype, which is death. It's important to all of us. But primarily, we're interested in looking at the phenotypic and genetic associations between education, intelligence, and their effects on health outcomes over the life course, including, or most importantly, cognitive decline and dementia, and also longevity. So, just a very brief slide, but education is very appealing in that it's relatively easy to measure compared to intelligence, and that you can take a fairly crude, self-reported years of education, or did you go to college, yes or no. Whereas intelligence, fairly time-consuming to measure, many different subdomains, so memory, reasoning, processing speed, so on and so forth. Different psychologists prefer different batteries of tests. It is, however, more granular and probably closer to the ultimate brain phenotype that we're trying to measure than education, which would be a more distal factor. With intelligence as well, typically when we have these different subdomains, we would use data reduction techniques like principal components analysis or factor analysis to create an overall general factor of cognitive ability. Importantly, though, both education and intelligence correlate with a heap of different lifestyle variables and health outcomes across the life course, including mortality, and in the expected direction as well, such that increased education and cognitive function associated with longer, healthier, happier lives. Maybe not happier, sorry. Longer, healthier. That's, yeah, sorry. Starting to feel the jet lag. If I do fall asleep, can someone wake me up? So the GWAS, well, we've already covered the education, EA2 GWAS today, just to say that when we do create polygenic scores from this and predict into our UK cohorts, we've excluded the UK-based studies from the original GWAS meta-analysis, so ISU kindly reran them to avoid bias in prediction. In terms of the biggest GWAS study of longevity, this was published last year by one of my colleagues in Edinburgh, Peter Joshi, and it was looking at parental lifespan, so something that was alluded to in the previous talk. So in order to maximize the number of events, so even kind of the most cold-hearted statistician, when you have a longitudinal cohort, you don't really want all your participants to die within a couple of years. And typically you'd have to wait 20, 30, 40 years before you get a large number of events in a cohort. Whereas if you look at parental phenotypes, so specifically take UK Biobank an example, so participants aged 40 to 70, many of the parents of those individuals will have passed away. And what Peter did in this study was a GWAS, so I suppose again, two ways you could think about it. So if you had either the parental phenotype is a proxy for the offspring phenotype, or in this case, we're saying that the offspring genotype is a proxy for the parental genotype. So here what Peter showed was that there were age and sex-related effects on longevity, such that on the left-hand side we have the GWAS for fathers in UK Biobank, so fathers' longevity, and showing that there's a nicotine-related gene that was linked to reduced longevity, and that had the greatest hazard in kind of midlife, and that reduced into late life. And then the opposite thing for mothers, whereby it was the ApoE allele, which showed stronger effects on mortality in the oldest age range of mothers. So what we did for our paper, which was published last year in PNAS, was to look to see if there's an association between the polygenic score based on EA2 and parental longevity. And just to note there that, again, we're maximizing the number of events by looking at this parental phenotype. So we did this in 3 different cohorts: UK Biobank, Generation Scotland, and the Estonian Biobank. And just an example, if we take Generation Scotland so the contributing number of participants to this study was 17,500, the number of observed deaths in individuals in Generation Scotland is 300 or 400 to date, whereas it's, I think, 6,000 or 7,000 of the parents of these participants have passed away. So UK Biobank, we've already heard a lot about. Just kind of skim through this. I've kind of copied here my favorite question from the verbal numerical reasoning test, which is, if Truda's mother's brother is Tim's sister father, what relation is Truda to Tim? Any thoughts on that one? And you had to answer 13 of these questions in, I think, 2 minutes. This is one of the harder ones, but yeah, it's a pretty tricky question. No one volunteering? Okay. So that's prefer not to answer then. So it is one of the options. So Generation Scotland, you might not have heard about. So this is a cohort similar to UK Biobank in that there's regional sampling across Scotland. Data collected at baseline between 2006 and 2011 on 24,000 individuals. So initially, just over 6,000 probands aged 35 to 65 were recruited through their general practitioner, and then they were asked to recruit their family members to the study as well. So wide age range of 18 to 100, 5,500 families. We have GWAS on 22,000, cognitive function on 22,000, and recently I've been working on EPIC chip methylation, which we have on 5,000 of the cohort. Then the Estonian Biobank, which Tonu can tell you an awful lot more about than I can, but I think as has already been mentioned, so it's representative, or 5% of the population of Estonia are currently participants in the cohort, and we had just under 8,000 contributing to this study with genotypes and parental phenotypes. So again, just to emphasize the benefits of looking at parental phenotypes, these proxy phenotypes. So these are the descriptive statistics for the mothers and the fathers of the participants from Generation Scotland Biobank and the Estonian Biobank. And we can see that of the participants in UK Biobank, 70,000 there were 70,000 events in the mothers, and 85,000 of the fathers were dead. So a far greater number than you'd expect from the actual participants themselves. And again, kind of mid to late life age range across all three cohorts. So as I mentioned, we took the polygenic score from ISU's paper, including all of the SNPs. And again, the polygenic score, kind of slightly lower estimates, lower R-squared estimates, around 1% of the variance explained in UK Biobank, and in the direction of higher score, more years of educational attainment. We excluded premature deaths, so those younger than age of 40, so primarily to try and rule out deaths by accident and also deaths from the Second World War, and then ran a fairly trivial analysis, just a Cox model of time to event in the mother, time to the event in the father, for against the polygenic score in the offspring, plus PCs to account for genetic stratification. And here we have purely illustrative plots, so just the polygenic score, EA polygenic score on the x-axis by decile, and of the participants' parents that died, we have the mean age of death within each of the decile bins, with the fathers in the dark colour and mothers in the light grey. And a few observations from this. So first of all, you can see that size is really important, where UK Biobank, you got those very tight error bars around the estimates. You can see, sadly, that life expectancy in Scotland, so the left-hand panel, is lower than UK Biobank. Oh dear. And also this kind of slightly funny bump shape in the Estonian Biobank, which could be a function of sample size, not entirely sure, but you see a fairly nice monotonic increase in the life expectancy in the UK Biobank, which is by far and away the biggest cohort. And this translated into around about a 0.6 year of increased life for those in the 66th percentile compared to those at the 33rd percentile. So a non-trivial increase in life expectancy, especially as this is probably half the effect of or half of the true effect, given that we've got the proxy genotype. So more formally, from the Cox regression models, we see that the hazard ratio is below 1, so suggesting that the increased polygenic score for education associated with increased longevity or decreased risk of mortality, and fairly consistent point estimates across the three cohorts. So with apologies to Dan, we compared the effect size here to the effect sizes for other polygenic scores per standard deviation. And we saw, so taking the results from various consortia projects, so Giant, Cardiogram, PGC, and TAG, we see that the effect sizes for education were stronger per standard deviation than they were for the other polygenic scores. So increased polygenic score for BMI, resulting with an increased mortality risk. So everything in the expected direction, that kind of hazard ratios, kind of 1.02 being the biggest one there. So associations with cardiovascular disease, nothing with MDD, major depressive disorder, and smoking associations only in the fathers. So in addition, we also looked at phenotypic educational attainment in the offspring in relation to parental longevity. And the hazard ratios for both the mother and the father were pretty much 0.9, so greater in magnitude per standard deviation than they were for the education genetic score. We also ran LD regression on the GWAS from Peter Joshi's paper, and that was on the a GWAS of the martingale residuals from the Cox model of maternal and paternal longevity. And the LD regression was with the EA2 output. And we see fairly sizable genetic correlations in line with a lot of other genetic correlations between EA and other health and anthropometric traits. In terms of additional things we looked at, so one of the reviewers suggested that there was potentially an issue by selection bias by having more children, you're more likely to be included in the study. So we, and for those that had more children, there may be a bias in the polygenic score. So we adjusted for number of SIBs in the model, which made no difference to that was only in UK Biobank, but made very little difference to the hazard ratios. And similarly, we adjusted for social class in the model, and that again made very little difference to the findings. One assumption would be that so people that don't have any children obviously can't be included in this study, so whether or not the effect sizes would be similar for them in terms of education on their longevity is just an assumption we'd be making here, but yeah, I suppose can be tested in future cohorts. So what does it all mean? So many potential pathways here. So firstly, we suggested that it might be a case that these education-related genes have some sort of biological effect on the brain, then increasing IQ, potentially motivation and conscientiousness, leading to more years of educational attainment, leading to an increase in quality of life, better environment, increasing to, or linking to longer life. And that could either be a direct effect through the pathway, or maybe, so of the parents with longer lifespans, or their offspring with higher educational attainment genes having better resources in midlife, so being better able to look after their parents in their old age and affording better health care. Another possibility is that we have health effects on educational attainment, health effects on longevity, and those are confounding the association between the education genes on longevity. And finally, the biological pleiotropy, such that the genes linked to educational or associated with educational attainment are also linked to health, physical health, neuronal health, and the multiple possible pathways through to longevity. So in terms of next steps, one thing we're going to look at is so the overlap in these different polygenic scores. So how does, in a multivariate model, if you include the education score, a BMI score, so on and so forth, do you get a better prediction of longevity? Do the findings generalize across other ethnicities? We may also be underestimating the effect sizes by something that has been alluded to already, so the selection bias in UK Biobank. We don't have information on parental education, which would have been nice to include in the model as well. And we will, of course, be running these or similar analyses to these in the full UK Biobank cohort once those data are available. So in terms of people to thank for this, in particular, Stuart Ritchie, who's joint first author, and Peter Joshi as well, and the other colleagues in Edinburgh. And the paper is published in PNAS last year, as I mentioned. So some other results that didn't make it into that paper, mainly due to sample size in the discovery GWAS, we're looking, again, same sort of thing, so polygenic scores for intelligence, educational attainment, and socioeconomic status, and seeing if they're predicting parental longevity as well. And we know that phenotypically these variables are associated with health, social mobility, and so on. So here we took UK Biobank as the discovery cohort, and then we were predicting in Generation Scotland only. So we took the verbal numerical reasoning measure in Biobank, GWAS that, 30,000, college versus no college GWAS in Biobank, 112,000, and then a GWAS of the Townsend Index, which is an area-based socioeconomic status measure in Biobank. Applied that to Generation Scotland, where the phenotypes we had available were this general factor of intelligence based on 4 cognitive tests. We had the Scottish Index of Multiple Deprivation, which is another area-based measure of deprivation, and educational attainment. So here we have the genetic correlations below the diagonal and the phenotypic correlations above the diagonal for these 3 measures, and kind of in the direction you'd expect, and of similar magnitude to those that have been published previously. So genetic correlation between education and intelligence around 0.7, phenotypic correlation around 0.3. So looking at the association between these polygenic scores within and across traits in Generation Scotland, we see that the education score based on Biobank explaining 1% or 1.2% of the variance in intelligence, the education score explaining 2%, and the socioeconomic GWAS score explaining 0.65% of the variance in socioeconomic status in Generation Scotland. And just on the final slide here, this is, again, analogous to the figures I showed earlier. So this is taking the GWAS results from Biobank, the polygenic score deciles, and then looking at the association with longevity. And not quite monotonic increases, but kind of nice trajectories here from the lowest decile to the top decile, so seeing that increase in life expectancy by polygenic score for intelligence, education, and SES. And then taking a principal component of the 3 polygenic scores, we see again a similar trajectory. So with that, I'll finish and take questions in 20 minutes' time.
All right, so I'm Jonathan Beauchamp, and I'll be talking about a paper entitled Genetic Evidence for Natural Selection in Humans in the Contemporary United States. Oh, there is one here. Good. Okay, good. So to get started, a few motivating facts. So first, until recently, it was often held that human evolution had come to an end about 40,000 to 50,000 years ago. So Stephen Jay Gould, who was an evolutionary biologist at Harvard and also a famous public intellectual, famously claimed that there has been no biological change in humans in 40,000 or 50,000 years ago. Now, privately, many biologists disagreed with Stephen Jay Gould. However, the conventional wisdom remained that humans had essentially not evolved in the last few tens of thousand years. Now, over the last decade, and since the advent of the Human Genome Project, evidence from so basically there's been lots of evidence that has accumulated that natural selection has been operating in humans over the past few thousand years. So for instance, adaptations such as lactase persistence, resistance to malaria, adaptation to high altitude, and the alcohol flush reaction in East Asians have been documented. And in fact, the precise genes underlying each of these adaptations have been identified and documented. As well, it's been shown in Europeans that there's been that height and body mass index have been under selection in Europeans, okay, in separate papers. So that really basically proves that the earlier prevailing conventional wisdom of no evolution was wrong. In addition, but this was from several thousand years ago, so these are adaptations from several thousand years ago. Now, recent studies have also examined the association between lifetime reproductive success, which I'll abbreviate LRS during this presentation, and various phenotypes. And these studies have found that natural selection has been operating in contemporary humans. Okay, so then the question, however, these studies that looked at contemporary selection, basically selection in contemporary societies, they analyzed phenotypic data set, so just with phenotypes. But natural selection only occurs when the genotypes associated with the phenotypes covary with fitness. So basically, to make the inference from these results with phenotypes that natural selection has been occurring, we need to make a few assumptions. Now, recent advance in molecular genetics makes it possible to look directly at the association between genotypes and lifetime reproductive success. I should say also lifetime reproductive success is essentially the number of children one has, okay, during a lifetime. Okay, so, and there's been a paper by actually Felix Tropf and Melinda Mills and a few co-authors that looked at the association at the genotypic level between number of children and age at first birth. Leveraging these recent advances. And then in this paper that I'll be talking about, I also leveraged these recent advances in molecular genetics, polygenic scores essentially. And I study the paper is entitled Genetic Evidence for Natural Selection in Humans, the Contemporary United States. It was published in PNAS last year. And as the title suggests, essentially I show that there's genetic evidence for natural selection currently. And essentially what I'm doing is I look at the association between polygenic scores and lifetime reproductive success for a number of phenotypes. And I find that some of these are significant. And then I derive this, I extend this to see what this implies in terms of rates of natural selection. Okay, so now for the data. The data comes from the Health and Retirement Study, or the HRS. And there's several reasons why this is a good data set to study this. So first, the HRS was designed to be representative of the US population over the age of 50 years old. And that makes it possible to generalize my results to the entire US population of European ancestry born in the years of my study sample. Second, individuals in the study are in the later stage of their lives. When they have typically completed reproductions. That allows me to study individuals that have completed reproduction. The study sample for the study that I use for the analysis is the set of unrelated individuals of European ancestry. I use these only to mitigate the issues related to population stratification. I use also individuals only born between 1931 and 1953. And there are some earlier cohorts in the HRS, but I found that there was evidence of mortality selection for them, so I excluded these earlier cohorts. And I also only use individuals who are at least 45 years old for female and 50 years old for male when asked the number of children they ever gave birth to or fathered. Again, the reasons why I limit the sample for individuals who are that old is to make sure this is completed reproduction. Now, I also do a robustness check, and the results are robust to using females that are 50 years old and older and males that are 55 years old and older. Now, the main dependent variable is relative lifetime reproductive success, or RLRS. That's how I'll abbreviate it during this talk. And RLRS is the ratio of LRS, or lifetime reproductive success, to the mean LRS of individuals of the same gender, born in the same years. And again, to remind you, lifetime reproductive success is essentially just number of children an individual ever had or gave birth to or fathered. So essentially, the dependent variable we're studying here is the ratio of one's completed reproduction to the average reproduction of people of the same gender in the same birth cohort. And the reason we use RLRS, or relative lifetime reproductive success, as opposed to just LRS, is because this allows us to translate the estimate into rates of natural selection, as I'll talk a bit about later. Now, the independent variables of interest are 7 phenotypes and their polygenic scores. There's BMI, education attainment, or EA, fasting glucose concentration, or GLU, height, or HGT, schizophrenia, SZZ, and plasma concentration of total cholesterol, which I abbreviate TC, and age menarche, or AAM, for females. And the control variables are birth year dummies, HRS-defined cohort dummies, and the top 20 principal components of the genetic relatedness matrix. Oh yeah, and I should mention, for the dependent variable, ideally the dependent variable we'd like to have is fitness. But fitness, biological fitness is, as has been said of it, is this concept that all biologists understand what it is, but none is able to define precisely. But in any case, it's at a high level, what fitness captures is an individual's contribution of his or her genetic materials to future generations. Now, in modern societies, lifetime reproductive success is considered to be a good proxy for fitness. It's not perfect, and I'll discuss some caveats at the end. But it's because there's very low infant mortality and most people reach reproductive age, it's a good proxy for fitness, typically. Now, phenotypic evidence. Let's look first at the phenotypic level, what we find, okay? And then we'll look at the genotypic level. So first, at the phenotypic level, the HRS contains phenotypic variables for 4 of the 7 phenotypes, for BMI, education attainment, height, and total cholesterol. And for each phenotype, I simply regress RLRS on the phenotype and on the control variables. So essentially, the idea here is if you see, for instance, that taller people tend to have more children, well, if height is also heritable, this suggests that height is under selective natural selection and that it's positively selected for, okay? And here are the results. Here are estimates from separate regressions of RLRS on each phenotypic variables for all individuals. Let's focus on the panel for female here. And so these are all separate estimates from separate regressions. So what we see is for BMI, it seems that stouter females tend to have more children, and less educated females also tend to have less children, as well as smaller females. So that suggests if these associations are also true at the genotypic level and not just at the phenotypic level, this would suggest that natural selection is operating on these. For males, we find similar effects, except not for height. Okay? And I should say these estimates are consistent with the literature on phenotypic association between phenotypes and lifetime reproductive success. So here I'm reproducing stuff that's been already documented. Okay, now to look at the genetic evidence for natural selection, I constructed polygenic scores of the 7 phenotypes. So I used the sum stats from the latest GWAS for each phenotype, and all the sum stats are from meta-analysis that exclude the HRS, so I made sure that this was the case. And the scores are constructed with LD-PRED using the individual's genotype SNPs, and the results are also robust using the Plink scores with all genotype SNPs. But today, the results I'll show you are from the LD-PRED scores. And here are the R-squares of the polygenic scores for various phenotypes. So in blue first, these are the R-square from the previously reported R-squares from the GWAS, whose sum stats I use. And in gray, we have the R-square that I estimated myself in the HRS. As you can see, and the R-square estimated in the HRS is defined as the incremental R-square. Well, it's basically from, we first run a regression of the phenotype on the control variables, which are birth year dummies and gender and the principal components, and then we add the score and we look at the incremental R-square. And so basically the incremental R-square that I get in the HRS for BMI is around a bit more than 0.8. For education attainment is about 7%. And here the difference between the blue and the gray bar is because the blue bar is from EA2, where we did not include the UK Biobank in the GWAS for this score. And here, for this score, I was able to include the UK Biobank, so we get higher power. For height, the R-square is above is around 0.17. And for total cholesterol, it's just above 1%. Okay? And here, I don't show the R-squared for the other traits because they were not in the HRS. I could not estimate them. Right, now, so let's look at the genetic evidence for natural selection. So for each phenotype, I regress relative lifetime reproductive success on the phenotype scores and uncontrolled variables. So basically, I did the same thing as what I did for the phenotypes earlier, except now with the polygenic score. And here are the results. Here again are estimates from separate regressions of RLRS on the polygenic score of each phenotype. And for females, what we see is that the coefficient on the score of education attainment is significant and negative, and the coefficient on the score of menarche is also positive and significant, although it's just marginally significant at the 10% level. As someone mentioned, that's not really convincing. But however, I should say this one here is at the p-value is 0.002, which survives Marioni correction for 13 tests. For males, it's also significant for education attainment. And if we pool males and females together, then we get a very similar coefficient of minus 0.032, and the p-value then is 1 times 10 to the minus 5 when we pool males and females together. And yes, okay, so basically now this suggests, so basically how do we interpret these results? So to tell you how to interpret these results, I need to introduce two concepts, or rather one definition and one concept. So the first definition is a directional selection differential. So the directional selection differential of a trait is the change in the mean value of a trait due to natural selection in one generation, okay? Now, the Robertson-Price identity is an identity that's well known in evolutionary biology. It essentially tells us that the directional selection differential of a trait is equal to the genetic covariance between the trait and relative fitness, okay? So basically, in our case, our proxy for relative fitness is RLRS. So basically, it says that the directional selection differential of a trait is equal to genetic covariance between the trait and RLRS, okay? So based on this, this implies that, my estimates imply that natural selection has been operating on the score of EA, so on the score itself, basically, if you take the score as the trait of interest, natural selection has been operating on the score at rates of minus 0.022 for males and negative 0.022, again, the same estimate, but for separately for, for males and females in any case, which is equivalent to about 1 week of education per generation for both sexes, okay? Now, this tells us that basically that in the space of one generation, the score will decrease due to natural selection by the equivalent of 1 week of education for both sexes. However, we don't really care about rates on the score. As Dan mentioned earlier today, it's good to rescale these estimates. So basically, under some assumptions and some derivations that I show in the paper, it's possible to obtain directional selection differentials of educational attainment and not just of the score, okay? And I show essentially that this is equal to the directional selection differential of EA multiplied by the ratio of the irritability of educational attainment over the score, the R-square of the score of educational attainment. And based on this, my estimates imply that natural selection has been operating on education attainment at rates of negative 1.30 and negative 1.53 months of education per generation among US females and males of European ancestry, born between 1931 and 1953. So a few things here. So first, notice here I extended my results from my sample to the whole US population in this last line here. The reason is because I have a representative sample. I feel justified in extending these results to the entire US population that matches that sample, so subject to some caveats, of course, that I'll discuss later. The other thing I want to mention is these results mirror first findings from the phenotypic literature that has documented many times the negative association between education attainment and number of children, as well as two recent papers, also in PNAS. One of these papers was from Dalton Conley and Ben and Dan and Jason that are here today. And in that paper, they look mainly at trends over time, but they also document negative association between the score of education, attainment, and lifetime reproductive success. And the other paper is a paper by Augustin Kong and Kerry Stephenson from Iceland, where they replicate this finding, but in a much larger sample of Iceland. Okay. Now, we find that natural selection has been operating for education attainment and possibly also for age at menarche in females. But it's important to emphasize that the rates of selection I obtain are very low. So essentially, my results suggest that natural selection has been operating relatively slowly in comparison to the rapid phenotypic changes over the past few generations. So for instance, the mean level of education attainment increased by 6.2 years for native-born Americans born between 1876 and 1951. So that's basically 2 years per generation. That compares to my estimates of negative 1.5 months of education per generation. So basically here, there's been what I would call a cultural override or environmental override of natural selection, such as the overall effect has been a huge increase of education attainment. Likewise, for age at menarche, well, it has substantially decreased in contemporary Western populations, while I find suggestive evidence of positive selection for it. So again, the environment seems to be dominating by far here. So to conclude, a few caveats first. So first, relative lifetime reproductive success is not a perfect measure of fitness. I said that first, it's not a bad measure, but it's still not a perfect measure. There's two main reasons for this, at least. The first one is that it just compares the contribution, it just considers the contribution of one individual to the next generation in terms of genetic material. Now, what we want is the long-term contribution over more than one generation. So you could look at grandchildren or great-great-grandchildren, for instance, especially if there's a quantity-quality trade-off in children. The second reason for this is RLRS does not account for reproductive cycle. So essentially, for educational attainment in particular, individuals that have less education tend to reproduce at an earlier age, and individuals with more education tend to reproduce at a higher age, older age. So basically, this is not accounted for in this. The second important caveat is that it's not possible to translate my estimates into projected evolutionary changes over more than one generation. Now, there's at least two reasons for that. The first one is a technical one. Technically, if you want to do projections over more than one generation, you need to know the genetic correlation between, you need to essentially know the rates of selection for every other phenotypes that are genetically correlated with the trait of interest. I won't go into details, but basically that's the case. The second generation is simply that culture and environment change very rapidly. So the rates of selection I estimated for people born between 1931 and 1953 are unlikely to be the same as those for subsequent generations. The third caveat is that my results, my study sample may not be fully generalizable to the US population of European ancestry born between 1931 and 1953. There's several reasons for that. So first, there's been mortality selection such that the HRS, so I look at people who have survived at least to age 45 or 50 for females and males. Now, among that generation, about 10% of people, according to US Social Security data, did not reach that age. So immediately I don't account for this. Second, not everyone has been genotyped, although comparing the results for the phenotypic data between the subset of genotyped individuals to those who have not been genotyped doesn't seem to suggest any difference, but still. So keeping those limitations in mind, my results strongly suggest that genetic variants associated with education attainment have slowly been selected against in my study sample. The rates of selection are very slow versus rapid increase in EA that are likely driven by cultural and environmental changes. And my results also suggest that age at menarche may have been under selection in females. And then future research can attempt to replicate my results by using even more representative samples and more precise scores and alternative methodologies. And on that note, actually, the results presented by Melinda earlier today, she also finds a documented negative genetic covariance between fertility and education, which is compatible with my finding. And thank you. So who's taking the questions?
You guys are in charge.
Okay. I think Jeremy was the first hand I saw.
Yeah, John, I think this is really interesting. But I'm kind of trying to get my head around what. So, in your way of thinking, would you call all changes in gene frequencies natural selection? Right.
Yep.
So just the answer to that would be yes. So
No, not all change in gene frequency. So for instance, there's other mechanisms than natural selection,
Sure.
such as genetic drift and population bottlenecks and all. So natural selection is essentially when reproduction co-varies with genetically co-varies with a trait. Okay. So essentially that's the.
Yeah, so I mean, I guess the question would be, is there any causal claim about, so I would normally, I guess, think of when you talk about a trait being selected for, that the variation in that trait had a causal role in the differential fertility in this case, in the differential fitness. And it's hard for me to see, and I don't think it's just sort of being resistant to the idea of genetic change, but the idea of a selection argument without an argument for the trait itself being selected on, or evidence of population change in the trait itself, right? So if anything, the trait, as you note, is going in the opposite direction in the population. So I guess the question would be, in what sense is this natural selection? Does that make sense?
Yeah, I think so. I would say it's fully natural selection in the sense that natural selection is blind to the actual, like basically for the definition of natural selection to happen, it's like the mechanism is not really relevant. Like it's just, so it could be, and by the way, I'm not claiming that education is being selected against here. It could be that other traits, like some personality traits or cognitive traits or whatnot, are selected for that are correlated with education. So what I show is that genes, genetic variants associated with educational attainment have been under selection. So I'm not sure that addresses your question, though.
Isn't it a definitional?
Yeah, it's a matter of, in part it's a matter of definition, yeah. Actually, another thing I should say is often people say, well, how can there be natural selection against education? You know, education is meant to be desirable. Well, actually, again, it's a matter of definition. It's not because, you know, natural selection doesn't really select for traits that some people deem to be more desirable than others or not. Like, you know, it's just a matter of definition, yeah.
So,
Okay, there's 3 in a row there. So from back to front.
So just to kind of follow up on Jeremy's question as well, you talked about Dalton and colleagues' other work, and I don't mean to speak for them, but as someone reading both sets of studies, I mean, the coefficients are almost identical, right? But especially from the Kong et al. and Dalton et al.'s piece, like negative 0.06, right, on number of children ever born. But the conclusions are radically different. Right? And the one thing that I noticed is, as far as the big difference between Dalton and his colleagues, is that they noted that the variation, right, the variance in these polygenic scores is stable over time, right? Whereas the mean value of polygenic scores is differing, but that variance is stable. And from my reading of it, that was a big conclusion that caused them to say that natural selection was not occurring. Right? So I'm just curious as to if you looked at variance, or if you didn't, why not? And just if you could enlighten us.
You mean variance in the polygenic
Yeah,
score?
correct.
So I did not look at variance in the polygenic score simply because, well, I mean, I did in the R squared is kind of the way I looked at it, where basically it's the share of the variance in the trait that's, it's like, the R squared of the score is essentially the variance of the score over the variance of the trait. But I did not look at trends over time. And I don't recall Conley at all stating that there's no natural selection. Several quarters in the room.
Well, we use EA1, so in some ways it's kind of a...
Oh yeah, that's true. One big difference is they use the first GWAS educational attainment group, yeah. Yep.
yes. So do you want?
How did, if the effect was almost identical, right? So Conley et al., that you found a negative 0.065. Kong et al. Found a negative 0.065. So the coefficient was almost identical. Does that matter then? It uses a different
That's the-
polygenic
that's the-
EA1 versus EA2?
What coefficient is that exactly? That's what
It's
is the
the
notation?
polygenic score of educational attainment on number of children ever born, right?
So it depends on the standardization of the score, though. So I'd have to check to compare that they use the same.
So this is more or less the question I asked before, but I'm puzzled by the schizophrenia results. So schizophrenia is same more or less as quite educational attainment, has a stronger effect on reproduction than educational attainment phenotypically. Why we don't see an association between the polygenic score of schizophrenia fitness?
Yeah, so two possible reasons I can think of right off. So one is that we simply are not well-powered in the association between, basically the association between the score of schizophrenia and reproductive success is weaker in 40 patients. It just might be a matter of power. Another possible reason is that, yes, phenotypically we do see that schizophrenic patients have lower fertility, but that's just like, what is it, percent of the population or something? A small fraction of the population is schizophrenic. So now this is looking at the variation across the whole population. So it could be that like being higher on the genetic score for schizophrenia doesn't really matter unless you actually are schizophrenic, unless you really are at the...
There might be some desirable characteristic of the schizophrenia spectrum.
Mm-hmm. Yeah.
Or it just doesn't matter. I have like a bit further, I mean, you did speak about this fitness with biologists, and then some people like this lifetime theory, that there are some people who just live fast and get more offspring, and other people live slow and get less offspring. Is there a way to like tease this out and see how different strategies play out in the long term?
So, yeah, I don't think I can get at that with this precise analysis. I think it would be interesting to look at this, but I don't think this, that allows to tease this. Yeah.
I have a question for each of you. On the selection, I'm kind of worried about selection on the 15 principal components and whether by orthogonalizing to that, you're actually leaving out some important aspects of selection. And on the other paper, it seems to me you could get a lot more power if you included not just parents, but you're going to get more power from the death dates and the grandparents, because they'll be almost all dead. And if you add across all 4 grandparents, you've got just as much genetic correlation in the uncles and aunts and everything. So I think one of the big virtues of being willing to look at people who are just genetically correlated rather than the person themselves is you might actually have a lot of relatives.
Do you want to
Yeah,
first?
so I guess it's data availability would be one of the key things for that, and biobank and the other cohorts we have in terms of just not knowing relative or other relatives other than the parents. So it's, and I guess you also have the reduction in like being a 50% proxy, their offspring genotype for the parental, but then if you going further and further, it's
grandparents put together. Well, I guess I don't know how these biobanks are put together, but if you can go in an enclave and know somebody's name, there are amazing genealogical things out there that you can look up people's death dates and birth dates and how many kids they had and so on. So it, I mean,
I guess that's an
sometimes
ethical
it can be a lot
question
of
for
work, but as long as somebody gives you access to the actual name of somebody in some top secret enclave, you can look up lots of stuff on their ancestors.
I think Joe has thought a lot about this problem from a theoretical point of view. I don't know if you have to it sounds clever to me that grandparents possible.
Yeah, so I mean, I think it's definitely a case of
And before I let you speak, I said Joe had a wonderful paper called Case Control Studies Without Cases.
Yeah.
And
we just maybe change the name of the paper.
but they made him change the name of the paper before it was published.
And so we look at this from sort of theoretical point of view, but and yeah, that's absolutely the case. From a practical point of view, there are a couple of interesting sources of data. There are things like death records in the United States are public, right? So if you could get all the death records, depending on the state, and start matching people to that would help a lot. There are things like the commercial companies, Gene.com, AncestryDNA.com, those people are actually making those family trees explicitly and attaching genetic data to them. So that is an extremely promising way to go.
And just to answer your question that was for me, so yes, it's a good point. I think the principal component explaining are quite weakly correlated with score and modification attainments. I don't think it's writing the results, but it would have been interesting to do a robustness check just without to see what's the differences.
go from left to right.
Yeah, I guess. Yeah, we'll go there and then. Who are the others who have questions? Matt and Nora. Okay, so Greg.
Yeah, just on this issue about going to the grandparents. In Britain,
that gradient between adult longevity and social status has become much sharper. So if you go to the 19th century, the gradient was actually very weak. And so if you go back to the grandparents, you actually lose, you get attenuation from that effect. And in fact, already by going to the parents, you probably attenuated it. But I think it's increasing over time.
I mean, that's assuming a constant relationship between EA2 and the social status of the individuals in the population, right?
Right. Wow.
And that may in fact not be the case in a more sort of class-based social system that you would find if you go back several generations in the UK, right? There may have been less opportunity for that kind of mobility, and as a result, you're going to get.
As a historian, I've got to stand up for the Victorians. It's a caricature, and to claim that somehow they had a more unequal society than our society. It's actually, it's very open and just mobile.
Yeah. Another thing that would have been lovely to have in Biobank was a measure of parental SES, but that wasn't assessed in the cohort.
So Matt and Laura, Matt first.
First a comment, then a question. In terms of the variance, in principle, of course, selection should exhaust additive genetic variance, but if you look at artificial selection, which is very powerful, there are studies of artificial selection on agricultural phenotypes that go over 100 generations, and they have not exhausted the additive genetic variance. So the change in variance, I think, is very, very slow. I wouldn't necessarily expect in yours. So Jonathan, some psychology has been very worried about dysgenesis for intelligence, not the same figure looking at for many, many years. And one thing that they've found to be very important in looking at potential dysgenic effects is whether or not people with intellectual disabilities are included in the sample, because they can have actually, it can completely change what's going on, and often people with intellectual disabilities aren't sampled. I don't know about the health retirement study. He talked about mortality selection, but are people with intellectual disabilities also included in the trials? Because that could really change things. It certainly, I would say there's data suggesting in the psychological domain that the degree of dysgenesis, if it exists at all for intelligence, can look quite different if those individuals are not in the sample.
Yeah, so actually the health and retirement study is designed to be representative, but I'm sure it hasn't succeeded in being perfectly representative. So that's almost certain that they miss some segment, or they don't represent every segment perfectly well. However, in terms of, I think Jonathan King wants to comment on this, but maybe you know better than me.
It's people you're sampling from households and PSUs. And if what you're talking about, a lot of the developmentally disabled adults are not living in the households, but are living in other places, you're not going to sample them. So I'm sure they're undersampled. But they're not ineligible for the study. The additional problem, however, may be in some cases, are they going to be willing to, or whoever's looking on, willing to let them answer the questions or participate by proxy interview, and that's sort of question. I'm sure they're undersampled, but no, they're not excluded.
It might be useful to probability weight things against some normative data, just to see.
You could.
Yeah, against the census, yeah.
Most of the things you can, we do.
Yeah.
But that's actually definitely going to be a case where I know that we're going to be undersampled.
No, you're right. Yeah. And I think Laura has been waiting for.
So thank you for the presentation. It's very interesting. And when I go back to that Joshi paper and look at the two GWAS, I really see, the guys smoked and killed themselves, and the ladies died of dementia. So you have these two really strong components there. And as we're kind of moving through this, how are we taking, can we take into effect? Can we take into effect some of the behaviors that we know really are correlated with educational attainment, such as smoking, and really are leading to increased mortality?
I guess it's a really or two very nice clean Manhattan plots with nice clean stories. Kind of, I suppose, a shame that they're so obvious, but it's kind of a nice confirmation as well that exactly. It'll be very interesting to see once the full genetic data come out to see if there's additional signals that kind of pop up where there's maybe some new biology that's.
But I think we have to take into account what we know and so it kind of goes back to this other question. Once we know something, can we take that into account and do our analyses to potentially uncover new things? Because
So-
those effects is so.
So adjusting for what, phenotypic smoking or something in
Yes,
the,
or something.
yeah. I can't remember from Peter's paper if they went and adjusted for that in the analysis, but it's,
Using polygenic risk scores,
Uh-huh.
and you could adjust for some smoking behaviors also.
mhm. Yes, absolutely. It's, yes, certainly something to consider. I think just as a shout out for DNA methylation, if you have methylation data, then that's almost better than as a proxy measure for smoking than self-reported smoking status.
Okay, so should we say 1, 2, 3?
Okay.
I just have a question. Did you, Jonathan, when you looked at how many self-reported children people have, was there a lot of missingness in that variable?
How many ways? Self-reported?
Self-reported children.
No.
Were there a lot of people who had missing data?
No, no. There were some, but I mean, no, in
But-
general, because
but
they,
there was a lot of bias there in terms of, because I know at least with parental education, the genotype sample, about 10% are missing parents' education. And it's not missing at random, but way lower genetic scores. So I'm wondering if there's a similar bias or maybe with self-reported children.
I'm pretty sure the answer is no, but I would have to double check. I mean, I checked these type of things.
Yeah.
So they were asked this question pretty much all waves of the HRS. So, and then, so at some point they usually answered. So there were like 10 waves.
So I was really interested in the fact that you control for the dummy effects of the period, I think. And I don't know how it is in the US, but some countries have dramatic fluctuations of fertility, especially in Eastern Europe after the economies have been fell by 50% or so. But I haven't quite understood the mechanism of that. I don't know if there's anything similar with the Second World War, baby boomers, and maybe there's some things to look at that issue, but have you thought about that at all? Like, can we use that in making an extra kind of analysis of differences?
Yeah, so actually I've looked at that, like basically in the paper I plot the average, and I use the phenotype, I use the dependent variable is relative lifetime reproductive success, so it normalizes by birth year average, and also use birth year fixed effects. Now, whether there's any additional inference that can be made with this, would you have any specific?
If let's say that in the 1950s, a lot of mothers had lots more babies, but you like control that out, so you don't see that. You're looking at the evolutionary effects that are happening beyond that.
I'm looking within birth year essentially.
Right. But if the mothers from the region have lots of babies, and they're having like different kinds of babies, then you're going to not see that. You're going to look at the kind of effects on top of that big wave. But maybe the big wave is really, really big. We should also look at the big wave.
Well, what I do is look at the effects in 3 different cohorts, essentially. So I split the period from 1931, 1953 into 3, and I looked at the effect in each, and they're quite similar. But
yeah, just to add to that, I mean, I haven't been keeping up with you've been doing the polygenic scores, so maybe you or Dalton's already done this, but I know in some of the twin studies we were doing a few years ago, and Melinda's done some as well, we were finding that heritability was increasing as a function of demographic transition. So during the sexual revolution, for instance, when people were using contraception more, and presumably taking more control over their childbearing as a function of their psychological phenotypes, we were seeing increased heritability.
infertility.
Both in terms of age of first birth and total number of children.
No, that makes sense. Yeah. I think if I were studying a very long time period, that would definitely be a concern. Because it's 23 years is not that short, but because it's still robust within periods, I'm less concerned. But yeah, it's a good point. I think David, you had.
Yeah, very quick as well, question on schizophrenia and PGS results you guys. I think that's exactly the kind of setting where Dan's proposal would make a lot of sense. So for height, BMI education, I think rescaling the estimate by H squared Smith over R squared would just cause a roughly proportional shift for all 3, but it wouldn't change all that much. But for schizophrenia and some of your other key types, it's really hard to know, right? And so one of your answers maybe is just power, and that you could immediately outfall from a set of normalized estimates. I think you should see whether there's a puzzled by for resolution of the schizo estimate, which is really, really imprecise.
Yeah, yeah. And it's true also that schizophrenia is more irritable than medication. So, I mean, that's what else. Yeah.
A quick question for Riccardo. Are the independent recall measures, or are they?
Yes, it's reported by the participants, yeah.
So recall is really tied to education, and so I wonder the extent to which there's selection across those that are able to recall precise ages of their parents to education, and certainly as a result of DCA, GS as well.
Oof. I guess it's a possibility, yes. Quite how you go about testing it.
Can you see whether they answered the question or not?
Okay, yes, I suppose that would be one where, yes, there are some, yeah, so we just removed, if there were missing data on parents' age of death.
I mean, for example, people are more or less precise
Yeah.
in their measurements.
Mm-hmm.
And there has been some work that looks at this in HRS, it's not genetic, but that could be a reference for the extent to which that.
Okie doke.
More questions.
Well done.